Okay, welcome back. We are now going to cover part of chapter 6, which is cell signaling. The last presentation I posted for you was finishing chapter 5 on membrane function, and now we're going to do chapter 6. I'm realizing that I really probably can't do the entire lecture. That would not be very fair to you, given that the last one was 40 minutes. And then to do another, half hour, 40 minutes would obviously be more than we would have covered in class on Friday. So I'm going to watch my clock and limit this to about 15 minutes, and then we'll finish it in class tomorrow. I reminded you, or I told you at the beginning of the last presentation that I included slide numbers so that if you have a question as I go through the presentation that you would like to ask in class on Monday, I'll give some time at the beginning of class that you can say, yeah, please go to, slide number 4 in the cell signaling lecture. I have a question there. And that way we can quickly find the slide where you had a question, and we can discuss that. Again, I also encourage you to pause the presentation at times and to think through material, particularly if I ask questions. Yeah, so pause it, kind of like we do in class, where I stop and have you discuss for a little while. Okay, we're gonna hop right in. So overview here, so cell signaling, this is what we're talking about. And we talked about at the beginning of this course about homeostasis and how the body responds to slight changes in its internal environment and then has mechanisms in place to quickly set those right when we're talking about negative feedback loops and maintaining that environment. And in order to maintain homeostasis and make all of that happen, then the body has to be able to communicate with itself. And that we have trillions of cells, and somehow communication has to happen between these cells. And that's what we're going to be talking about today, cell signaling. Okay, so signal pathways. By the way, I'm trying to do this audio a little differently so it doesn't end up garbled like it did last time. So hopefully hopefully it's a little better quality. Okay, signal pathways. So you can see over to the right of this slide, we have kind of the familiar looking circles, ovals, and rectangles that are color coded, and your book will try to be consistent in their use throughout your book to mean these particular things. So there is a signal that is sent. Likely that means release of some kind of molecule from a cell in your body. So it's made and then released by a cell. It's a signaling molecule. It has to be transported to target cells. What are target cells? Well, we'll find out what that is. It's not usually not every cell in your body, but particular cells that are going to respond to the signal. So signal molecule is sent, and then that has to be received, and that usually is by a receptor in the target cell, and that's kind of what actually determines what the target is who has the receptor for this particular signal molecule. And so we typically see this through the binding of these signal molecules, and we will call these ligands. And this is a signaling molecule, and it will bind with its receptor protein of the target cells, and that's actually what determines what is a target cell, is who has the receptor for this ligand. And then we have signal transduction and amplification, and so this is a conversion of the extracellular signal to an intracellular signal. I'm seeing a mistake on this slide as I'm talking right now. The first little bullet under number 3 that says signal transduction and amplification should say conversion of extracellular signal to intracellular signal. And then this typically involves a chain of molecules that's going to relay information and what we'll call amplify the signal through second messengers. This might not make a lot of sense yet, but hopefully it will by the end of this lecture. And then we get some kind of response. So the final molecule in this series of reactions, the final molecule is going to trigger a change in some cellular process. Okay, so that first thing, we're going to talk about that first, sending the signal. Okay, so we will see that we have local communication. This occurs over very short distances, and if you're thinking ahead, you might say, hey, the next thing she's going to talk about is long distance communication, and indeed I am. But right now let's talk about local communication. So this can happen in kind of 3 basic ways. So one, we have direct communication some kind of connection between the cytoplasm of cells that are right next to each other, of adjacent cells. And these are called gap junctions. And you see that bottom left under A there. And you see these, there are these proteins called connexin that penetrate both of the cell membranes, and it allows electrical and chemical signals to flow through those pores. So electrical meaning ions, right? Ions can pour through those pores. And as well as chemical signals. So this is kind of a classic example of this in the human body is going to be in cardiac muscle. So cardiac muscle cells are joined by these gap junctions, and this allows a very coordinated contraction of those muscle cells together. And so when you have that very rapid communication, it means they can kind of make things happen at the same time. The next one is contact-dependent signals. So here we have binding between surface molecules and receptor proteins. So notice there, that's the one in the middle there at the bottom, that the signaling molecule is attached to a cell membrane and the receptor is attached to another cell membrane. And so you have to have surface-to-surface contact between these cells for this signal to be sent. And then those, the last two, paracrine and autocrine signals. So we see down there at the bottom right, there's three little cells lined up, and the one on the left is making the signal molecule. And that signal molecule is going to affect the same cell that made it. That's called an autocrine signal, when the cell that made the signal is also going to receive the signal. But we also see that the signal molecule that it's making is diffusing a short distance and affecting other cells in the near vicinity, adjacent cells. And in that case, it's called paracrine signal. And you can also see in that example that you can see that a cell can be both autocrine and that it can be sending autocrine and paracrine signals. So that cell on the left is the one in this example that's making the signal molecule, and it is affected by it, but it's also affecting the neighboring cells. So in that case, it's going to be autocrine and paracrine. Okay, so here we've moved on to long distance communication. Get to the right spot here in my notes. Okay, long distance communication, and this kind of communication is going to take place either through the endocrine or the nervous systems. And so with the case of endocrine system, we have hormones. Those are going to be the signal molecules, hormones that are made by an endocrine cell. It's going to be secreted into the blood, and it's going to be carried throughout the body. And again, these signal molecules, these hormones, are going to come in contact with just about every cell in your body. But not every cell in your body is going to respond to these hormones. Only the cells that have the receptor that's appropriate for that hormone is going to respond. And so that is how we get the specificity, that only the cells that have the receptor are going to respond. And so yeah, okay, enough about that. Hormones persist for a longer period of time after they're released than paracrine or autocrine signals. So you might be asking, like, okay, what's the difference in these hormones and then the autocrine, paracrine stuff we saw on the previous slide? Well, one would be that these typically are put into the bloodstream and they travel long distances instead of short distances. But the other is that these tend to last longer and have longer lasting effects than autocrine or paracrine signals. Okay. Sorry, trying to make sure we were going. We are. Okay. So the other way of long distance communication is nervous system. And so here we see that an electrical signal is converted into a chemical signal. So what does that mean? So see the little neuron there at the top left, and there's an electrical signal that's traveling down the axon of that neuron. And what happens when that signal, that electrical signal, reaches the end of that cell, right? there are no gap junctions here, so we can't just have a continuation of the electrical signal. But when it reaches the end, then there's going to be a chemical released. So that's why we say it's converted an electrical signal into a chemical signal. And that chemical that's going to be released, we have a broad category. We call it a neurocrine molecule. And then 3 different kinds of neurocrine molecules. Probably the one that you are most familiar with and that you already know something about is going to be neurotransmitters. So neurotransmitters travel a short distance and they have rapid effects. So we think of this most often where a neuron is going to release a neurotransmitter into a little space, a tiny little space, and it's going to diffuse across that tiny little space and then have an effect on the next neuron. And in that case, it's then a conversion of a chemical signal back into an electrical signal, because it's going to, sometimes it's going to make that next neuron fire, have an action potential, and keep going. But anyway, neurotransmitter travel short distances, have rapid effects. And then we have these neurocrine molecules called neuromodulators, and they travel short distances, but they act more slowly. So for our purposes, that's enough to know, is that they act more slowly than neurotransmitters. And then we have neurons that can release their product into the blood, and it can circulate throughout the body. And we call those neurohormones. So just like with an endocrine cell, endocrine gland, they're making their signal and they're releasing it into the blood, and it's going to travel throughout the bloodstream to its target. And again, what's its target? The cells that have the appropriate receptor so that it can respond to them. Okay, so that was a little bit about sending the signal, and now we are going to talk a little bit about receiving the signal. So that means talking about receptors. So receptor proteins, we can divide them largely into these 2 big categories of lipophilic or lipophobic ligands. And remember that the phospholipid membrane, that it is very hydrophobic. Those little tails that face each other are hydrophobic, which means that they are lipophilic. So they don't like water, but they like lipids. And so lipid, and I mean they are lipids too, right? they're phospholipids. So lipophilic ligands can directly pass through the cell membrane. So they can diffuse through the cell membrane, and then they're going to bind to the receptors, which have to be inside the cell somewhere, because if this thing has gotten inside the cell, that's where its receptor is going to be. And this happens slowly. And we'll see kind of why in a little bit. But this, diffusing across the membrane and then having some effect inside the cell is going to be a little slow. I mean, on the order of, an hour or longer. And then we have lipophobic ligands, and those are going to stay outside of the cell. So they remain in that extracellular space, extracellular fluid, and they're going to bind to receptors that are on the surface of the target cell. And we see on the cell on the left, back to the lipophilic, we see that there's a receptor inside the cell that can either be in the cytosol or it can be in the nucleus. And it usually results in some kind of change in gene expression, gene activity. And that's why one of the reasons it tends to be slower. And on the right, we see that the receptor is on the cell membrane. It stays on the cell membrane. The ligand stays outside the cell as well, but interacts with its receptor there, and then that's going to set off some set of events, and it's going to usually result in a pretty rapid cellular response. Okay, I'm realizing that I'm pretty much already out of time. So in class tomorrow, on Monday, I'm going to pick up with receptor proteins, and we're going to talk about the types of membrane receptors. So I will see you in class on Monday, and be sure and ask any questions that you have on what I covered over the rest of Chapter 5 or what I was able to cover here in Chapter 6. Stay warm. See you soon. Bye.